In this subtopic, we've covered the process of sugar loading, and we are moving on to electroosmosis. Within electroosmosis, we shall look at our definition. Electroosmosis is movement of water along a charged membrane. We have our illustration, and within this illustration we have the subtube element. Then we have the companion cell, and we have our sieve plate there. The sieve plate is this boundary there. Then, within this theory of electroosmosis, the main areas of focus are going to be the process, evidences that support the theory, and the weakness of the theory. We have little to talk about here. And the first point we shall look into is 1. We know very well that we are looking at movement of sugar from the leaves. We are focusing on movement of sugar from the leaves, I will say up, and sugar is moving down to the stems. So our sugar is here. So this is our sugar moving from the leaf area, sloping down. But as this sugar is moving from our theory of electroosmosis, first it's going to encounter hindrance from the sieve plate here. But the sieve plate has sieve pores. So we shall call these ones pores. And this companion cell is actively pumping potassium ions from the lower sieve element to the upper sieve element. So it actively pumps potassium ions from down to up. As potassium ions accumulate within the upper sieve element, these potassium ions are positively charged. So they are going to cause a net increase in positive charges at the upper part. The positive charges are going to increase, or the positive charge increases at the upper part of the sieve plate, leaving the lower side negatively charged. As the lower side is getting negatively charged, the whole surface is going to be polarized. Such polarizations are evidenced in impulse transmission. So, as you see here, we have the positive charge accumulating up and the negative charge accumulating down. Therefore, as the membrane gets polarized, water and the solutes that are within, when I talk of solutes, I'm talking of sugar and some ions. So I'm talking about sugar and some ions within water are going to start moving down from the region of moving down from where the solution is positively charged to this side that has a negatively charged solution. Now as this water comes along with the sugar. So the theory of electroosmosis tries to illustrate that Potassium ions are actively pumped by the companion cells using ATP. So, using ATP. As the potassium ions are being pumped using ATP into the companion cell pumps them out into the upper sieve element. Then in the upper sieve element, these potassium ions, since they are positively charged, are going to increase the positive charge up as they leave the negative charge down. And with this negative charge, water, at this point I will call it a solution because it's serving sucrose molecules and some ions within it. So this solution is going to start moving from the positive side to the negative side. And as it moves like that, that's what will cause movement of our sucrose from one sieve element to another. So this process repeats throughout. The process is going to be repeating throughout the entire flow. So when you look at our theory, the theory we've tried to illustrate here, the potential is maintained by active pumping of potassium ions in an upward direction. That potential here is maintained by active pumping of potassium ions in an upward direction. A charged solution flows from the positive to the negative direction, and this charged solution is our water. So what are the weaknesses of this theory what are the weaknesses of electroosmosis? One, there is little experimental evidence that this happens. So this is electro-osmosis.